This is lecture RP1. In this lecture, we're going to start by talking about the overall purpose of the reproductive system and what sexual reproduction is for. We'll spend a little time going over the idea of chromosomes, genes, and alleles, and the process, cell division called meiosis and how it produces gametes, the concept of diploid and haploid. We'll then talk about the different levels of sex determination, including chromosomal, gonadal, anatomical, and psychological sex, as well as the most typical way those align. And then we'll talk about some of the ways that those cannot align in typical ways, what we sometimes call intersex conditions.
All right, let's talk about the reproductive system, which is the system of the body designed to do one of the most characteristic functions of living things, which is to make more of themselves, or more versions of themselves, which is really quite extraordinary. Now, there's more than one way that living things can make more copies of themselves. For example, most cells, when they reproduce, at least animal cells, reproduce by mitosis, which is a process by which you make you divide into two cells which are, barring mutations, exact copies genetically of the original cell. So you split from one into two, and those two have the same genetic code. And some animal species and other species reproduce whole organisms by a kind of a related method. They have asexual reproduction, where in most but not all cases, the offspring are clones of the original. They are genetically the same, minus a few mutations. There are some more sophisticated ways of doing asexual reproduction that involve a little bit more genetic mixing, but in general, asexual reproduction produces very little genetic diversity. You produce lots of copies with very similar genetics. Sexual reproduction, on the other hand, allows for a lot more recombination of genes. So you take two individuals with slightly different genetic codes and then create a new organism that has a new mix of genetics. And that has a lot of evolutionary advantages. By increasing the amount of genetic diversity in a population, you have more possible organisms, which means when changes come, more of those organisms are likely to have mutations or particular combinations of genes which make them able to adapt to that new change. That's one of the things about evolution that is often misunderstood. We sometimes think about evolution as helping a species come up with new mutations to deal with a change, but that's not actually how it works. Evolution is about having change select for mutations. You already have to have the diversity. You already have to have the new versions that will adapt better to the change. So sexual reproduction is a way of increasing genetic diversity in a population, which has big evolutionary advantages. However, it does get a little more complicated. So in order to talk about sexual reproduction, we have to talk a little bit about how chromosomes work. Now, in eukaryotic organisms, which is more or less everything except bacteria, most of those organisms are what we call diploid. And to understand the idea of diploid, we need to understand the idea of a chromosome. So in non-bacteria, the chromosome our DNA is arranged in multiple individual pieces called chromosomes. So a chromosome is one piece of DNA. These can be very long pieces, millions and millions of nucleotides long. And that chromosome will have areas on it which encode proteins, which contain the instructions for building proteins. They'll also have areas that are involved in controlling which proteins are made and other stuff. But when we talk about genes, we're talking about regions which include instructions for building proteins. So a gene is a sequence of DNA which contains instructions for a protein. So let's imagine that we have a chromosome. We'll call it chromosome 1. And that chromosome will contain 4 genes. So here's a region, gene A, B, C, and D. So this is chromosome 1, and it contains 4. Most chromosomes contain a lot more than 4, but this chromosome contains 4 genes A, B, C, and D. Now, a diploid organism, which is most of them, will actually have 2 copies of chromosome 1 in most of its cells. So we'll have another one here called 1'. And that version will have the same genes under most circumstances. So it'll also have gene A, B, C, and D. So for example, each of my cells contains 2 copies of each of my chromosomes. I got one from my mother and one from my father. And those chromosomes should have the same genes, barring some very unusual circumstances. But those genes are not necessarily exactly the same version of the gene. Those genes can be different versions. It's sort of like having 2 copies. If I had 2 copies of a book that had 4 chapters, but in one of the books the chapters were a different revision, so there were some typos corrected and a few sentences altered, it'd be the same chapter, but a slightly different version of the chapter. In the same way, for example, this chromosome 1', its version of gene A might be a little different than chromosome 1, so we'll color it differently. Shade it in to show that it's a slightly different version of the gene. Now, that different version, meaning it's a different sequence of nucleotides, can mean that it produces a different version of the protein, that the instructions are a little different. So it includes a different amino acid in one place, or it changes the sequence of amino acids, or even it changes it in such a way that it doesn't produce a usable protein, or it produces a fairly significantly different protein. Lots of different possibilities. But the idea is that gene A here has 2 different alleles. So alleles are versions of a gene. So this individual in their cells has 2 different versions of gene A, this hollow version and the shaded in version, 2 different alleles. We say that somebody whose 2 chromosomes have 2 different versions of the gene is heterozygous. So if I have on my mother's chromosome 1 version of gene A, and on my father's chromosome a different version of gene A, 2 different alleles, then I am heterozygous for gene A. On the other hand, they could be the same. Let's say these 2 versions of gene B are exactly identical. In that case, I am homozygous for gene B. All right, so we can have different versions of the allele, and how that comes out, that depends on the gene. If you've taken basic bio, you've done the Punnett squares, and you know all about the idea of dominance and such like. So we're not going to get into that right now. The idea here is, though, just to get the idea that in a diploid cell you have 2 copies of each chromosome, and while they should have the same genes, they may not have the same version of the gene. So your cells can contain multiple versions of each gene. Now, humans have 46 chromosomes arranged into 23 pairs. 22 of those pairs work like this. The last pair works a little differently. So we're going to talk about that in a moment. But when we're talking about the idea of sexual reproduction, I wanted to get to the idea. Where did I put my eraser? Ah, there it is. When we're talking about sexual reproduction, I wanted to get to the idea of how we produce a new diploid organism. So I'm going to erase this right now. So if each of my cells is diploid, I'll represent that like this, with having 2 copies of the chromosome. Actually, let's do them in different colors to represent that they may not be exactly the same. We call these 2 versions homologous chromosomes. Meaning they contain the same genes, although not necessarily the same versions of the gene, not necessarily the same allele. So if I've got a diploid cell, And I have another organism that also has diploid cells. So there's its two homologous chromosomes. And we're going to undergo sexual reproduction. If I just take one of my cells and combine it with one of their cells, I'm going to end up with a cell with 4 copies of the chromosome, something called polyploidy. And in humans and most other animals, that doesn't work. Things do not work properly if you don't have 2 copies of each chromosome. So in order to make this work with sexual reproduction, I'm going to have to make a version of one of my cells that has only 1 copy of the chromosome. This other individual will have to make a version of their cell that only has 1 copy of that chromosome. We call these gametes. Gametes are what we call haploid, as opposed to diploid. So my body cells are diploid, but when I produce gametes, they only have 1 copy of each chromosome, which we call haploid. And when those 2 haploid gametes combine, I end up with a new diploid cell called a zygote. That zygote has 2 copies of each chromosome, but they won't be exactly the same as either parent. They'll have a new mix of alleles. And this zygote can grow into a new individual. So, I produce a gamete, my partner produces a gamete, those gametes combine into a diploid zygote, and that zygote becomes a new individual. And thus you get sexual reproduction, a new mixing of chromosomes, so more genetic diversity in your population. This zygote will have some of its alleles from one parent, some from the other parent, but its particular mix will be unique. So that's the basic idea. Now, how we produce the gametes, that's another interesting question. I'm going to show you the general idea of the process, and then we'll talk later about how it gets specifically implemented in production of sperm and eggs, which are human gametes. Okay, let's talk about the general process of meiosis, which is the kind of cell division that produces gametes. It goes from a diploid cell to haploid gametes. So the general idea is this. We start with a diploid cell, and I'll draw this cell with a red chromosome and a blue chromosome. So, 1 chromosome, diploid, so it's got 2 copies of that chromosome, 2 homologous copies of this chromosome. Meiosis, in meiotic cell division, we're going to start by duplicating these chromosomes. So I will produce 2 of chromosome 1, so each of those versions of chromosome 1 duplicates itself. And these 2 pairs are going to associate with each other, and there's actually something really interesting that goes on at this point. As these 2 pairs associate with each other, they're going to twist, break, and recombine in such a way that I get some slightly different versions of each. So here, for example, what we did is have these 2 copies of the chromosome twist over each other, break, and recombine, so that now I've actually got I've got some chromosomes that are a mix of both of my 2 original chromosomes. This is an event called crossing over, and it's actually an important part of this. All right, so during this after the duplication of DNA, we end up with some chromosomes which aren't quite like any of the originals. Then, this is going to undergo a first meiotic division. We're going to separate those chromosomes into 2 daughter cells. And then each of those is going to undergo a second division. Where we then split those chromosomes into the daughter cells. And what I've got is 4 haploid gametes, each of which has 1 copy of that original chromosome. So we went from a diploid cell, 2 copies of the chromosome, to 2 cells, which whether you call these diploid or haploid is complicated. But in the end, you end up with 4 haploid gametes. Each of these could be a sperm cell or an egg. Now, how this is more or less how it works for sperm. Eggs work a little differently, and we're going to talk about that maybe toward the end of this lecture or the beginning of the next one. But that's the basic idea of meiosis, how we go from a diploid cell to haploid gametes. And note that each of these haploid gametes, its copy of the chromosome is a little bit different. So depending on which of these combines with the gamete from the other partner here, you're going to get, it's hard to predict exactly what genetic mix you're going to get. It's sometimes when people don't understand this, they figure, okay, so when I, say a male, produce sperm, each of my sperm should be genetically identical because it all came from me. But it's actually not at all. Not only does each of my sperm have either my mother's or my father's copy of one of the chromosomes, and then you do that for each chromosome, but the chromosome it gets could actually be a scrambled version of my mother's and father's chromosome. So the genetic diversity just in your gametes is enormous. Each sperm or egg is probably going to be a little bit different from most of the others. So kind of interesting just right there. A lot of genetic diversity, so what you get in the offspring of sexual reproduction really can't easily be predicted. It's going to be kind of like you, but it's going to be different, and each one you get from the same partner will be different, barring identical twins, which is a different thing. All right, so that's the basic idea of meiosis. Now, let's get into the idea of how this works in human beings. So, we've talked about gamete production, and I've been suggesting that some humans produce sperm and some produce eggs, and that is more or less true. But let's talk about how that goes in terms of human sex. So, not the process, but the idea of male and female. And our society places enormous importance on this. When someone says, hey, I just had a kid, one of the first things they get asked is, boy or girl? And in recent years, this has become a very controversial topic because, of course, some people say, I don't really feel like either, or my body is that of a male, but I feel like a female, or vice versa. Or lots and lots of possibilities. So I wanted to talk a little bit about how human beings go through sexual differentiation. Now, I think it's best to think about this as having 4 levels of determination. So we can talk about an individual's chromosomal sex. We can talk about their gonadal sex. We can talk about their anatomical sex. And we can talk about their psychological sex. And those 4 usually, but don't always, align. So each of these affects the ones downstream. Your chromosomal sex affects your gonadal, anatomical, and psychological sex. Likewise, your gonadal sex affects your anatomical and psychological, and so on. They build on each other. And there are some typical ways in which they align. But the ways that are not typical are not abnormal, they're just unusual. So, for example, you will sometimes hear people talk about sex determination is very simple. There are 2 chromosomal sexes, XX and XY. We'll talk about what those mean in a second. And XX always produces female all up and down the line, and XY always produces male up and down the line. And that's the way it is, and it's simple biology. And I will tell you, as a biologist, it's not that simple. That is the most common, the most typical way things line up. But it is not the only way it can work, and we understand some of how the alternatives work. So, it's not that easy, and I'm going to talk to you a little bit right now about how it usually works and why it sometimes doesn't work the typical way. So, let's start with the idea of chromosomal sex. Okay, chromosomal sex. So, human chromosomes. Humans have 46 chromosomes in our diploid cells. Those were arranged in 22 pairs of what we call autosomes. So autosomes are sort of what we think of just the regular chromosomes. Those always, or at least almost always, come in easy homologous pairs, where you have 2 copies of chromosome 1, 2 copies of chromosome 2, and so on down through chromosome 22. And then you have 1 pair of sex chromosomes. And this pair is not necessarily homologous. So we're going to ignore the autosomes for now and just pay attention right now to the sex chromosomes, because those are the ones that are important in sexual determination. So everybody has an X chromosome. That one I can say, because if you don't have an X chromosome, you don't have a viable individual. One sometimes thing that people misunderstand about this is that they think that, okay, the sex chromosomes contain all the genes that have to do with reproduction. Not the case. The X chromosome is actually a large chromosome that has genes that have nothing to do with reproduction. It has a lot of genes that have to do with everything else that goes on in your body. Actually, the X chromosome, well, I mean, it has some to do with reproduction, but not a whole lot more than some of the others. But anyway, it's an important chromosome on its own. So everyone has at least 1 X chromosome. Some people have 2. But then there's also the other thing that can be one of the members of the sex chromosomes is the Y chromosome. The Y chromosome is a small chromosome that only has a few genes, none of which seem to be critical, because some individuals don't have a Y chromosome and their bodies work just fine. There's one gene in particular on the Y chromosome of interest. I'm going to note that right now. The SRY gene. We're going to come back to what that is. So, typically, an individual will either have 2 copies of the X chromosome, 2 homologous X chromosomes, or they will have an X and a Y chromosome. You always have to have at least 1 X chromosome. Your second one of this pair, if it's a pair, there's other possibilities, will either be an X or a Y. The typical pattern, if you have 2 X chromosomes, is that you develop female characteristics. Most individuals that are XX end up with ovaries, end up with female genitalia, female secondary sex characteristics, and a female gender identity. Most individuals with XY end up with testes, produce sperm, have male genitalia, male secondary sex characteristics, and male gender identity. That's the most typical way. It's not the only way, but so when people talk about chromosomal sex as XX and XY, this is what they're talking about. If the individual has 2 X chromosomes, typically you get female sex. If it's XY, typically you get male. But it's not that simple, and it's not really a binary thing. It's not necessarily one or the other. So, we're going to talk for a moment here about what it is about this chromosomal sex that affects sex determination, and how chromosomal sex interacts with the other layers of sex. In order to do that, we've got to kind of go over how this whole thing works, the gonadal and anatomical differentiation. When we're done with that, we'll come back to this and talk about what happens if it's not just an XX or an XY with an SRY gene. What happens if it's some other variation? So, we're going to come back to that, but that's the basic idea of chromosomal sex. Now let's get into how chromosomal sex affects everything else.